Hello, I am Enes Sefa. I am from Koç University. I'm doing my master degree in Network Modeling Research Group. Today, I will talk about my master thesis project and my poster presentation, which is titled as Leveraging the Molecular Signatures of Cancer for Dynamic Network Modeling. So in this study, let me give you first the outline. I will talk about the direct resistance, background information about the biological question, and then I will give information about my computational tool in the study I used. And then I will show you how I use this GCA method to implement the molecular simulations within the cell. And then I will give information also about how I performed the in silico perturbations. And then I will give workflow and the results. Direct resistance in cancer is developed within a very short time compared to the how cancer progressed over the very, very long time of the accumulation of mutations. We know that the accumulation of mutations leads to some abnormal activities in some cellular pathways, but in the direct resistance, we also see more than one or couple of alternative pathways that are showing the abnormal activity. Actually, how this works, with the cancer, we applied some drugs to cure the cancer, but some cells get resistant due to the drugs are actually putting a lot of stress on the cells, and this leads to increased mutation rates. And by natural selection kind of mechanisms, evolution-based mechanisms, cells accumulate a lot of mutations in a very short time, and some other pathways that affected and get resistant to the drug we are using. And also there is a supporting argument. There are some combinatorial therapies that when we block some of the pathways at the same time, we are able to overcome this drug resistance at some level. So in this study, I used actually the graph-based cellular automata. In this model, there are some target nodes and some neighboring nodes, and target nodes activity state at the next time step is determined by the activities of the neighbor nodes at the time step t. So in this method, we are able to synchronously update all of the node states at only one iteration. And then through the iterations, actually, we are able to model the dynamic nature of the molecular signaling. So in our model, we have directed interactome, which is composed of several different databases, and it's a heterogeneous approach. We have some gene regulatory networks, we have some protein-protein networks, and we combine all of them in just one model. And here we have some stimulations and some repression information, again coming from other databases. So we are trying to actually predict the activity level of the target node Y at the next time step based on considering all the neighbors. And we are simulating this molecular signaling without the mutations. And then we are applying in silico perturbations and deactivating some of the edges and comparing these two networks through the iterations and trying to come up with dynamic nature of the cause of the drug resistance. Here is the estimator we used between the edges. For example, RNF11 is activating the H1 gene, and this is the expression distribution of the two genes, and each dot represents a sample. So when we look at the RNF11 activity level around like 4.5, we see that each one activity is normal distributed. And if we know that the RNF11 is stimulating the H1, so we make a prediction like 2 standard deviation above the mean, and vice versa for the repression information. If it's repression, we estimate below the mean. And in terms of the mutations, we have 2 types of mutations. In the first one, it's a deleterious mutation. When it happens, we deactivate all of the neighboring edges and the downstream edges in the same pathway. If it's a point mutation, we look at the interacting residues and we deactivate the specific edges and their downstreams in the same pathway or complexes. In our general workflow, we are able to simulate for each cell line the GCA model, and we get full stack of the networks that we can compare. And by using the drug resistance information, we can make a majority voting. For example, taking one drug and looking at the sensitive cell lines to this drug, we can perform majority voting and come up with nodes that are unique to the sensitive cell lines for this drug. And same for the resistant cell lines. We have resistant cell line specific subnetwork and the sensitive cell line specific network. And we have also used the patient's mutation profiles to come up with the patient's modules, let's say, subnetworks. And then we can compare these subnetworks within an interactome by looking at a distance. If the patient module is more close to the resistance module for the drug, we can predict it as a resistance for this patient, and the same for the sensitive actually. In the results section for each drug, we have two sets of consensus subnetworks. One is the sensitive subnetwork, and one is the resistance subnetwork. And for the patient subnetwork, we are comparing all of these networks and looking distance. And in this proximity analysis, we have also Z-score, which implies the significance of the actually proximity analysis. And as the Z-score gets smaller, our predictions become more accurate, but we are able to make less predictions. Thank you for listening.